Thank you very much. It's a great pleasure to be here, and I thank your principal, Dr. Tyagi, and all of you for hosting this extraordinary event over here. People normally say that a speaker needs no introduction because they're old and famous, but this speaker needs no introduction because he's young and full of impact globally. You can Google Manu Prakash and see what he's been doing, and it's absolutely stunning to see someone who was a kid growing up nearby, who studied in Kanpur and then went to MIT and is now a faculty member at Stanford, do science which is absolutely stunning. All of us growing up, when we read about the history of science, one of the things we read about is how people gazed at the stars, wondered about them, and understood by doing that the laws of physics, astronomy, and really changed the world in many, many ways. Interestingly, while we are inspired by the stars, and we are inspired by large life forms around the planet, including ourselves, elephants, tigers, dolphins, whales, and so on, we rarely turn the telescope around and look at small life forms around us. And that's what Manu has done. He's inspired the world to look more closely at itself. And this is really, really tremendous for three reasons. One reason comes from Charles Darwin and Alfred Wallace, who showed that the diversity of species on this planet is a common origin and has diversified by natural selection. Now this is phenomenal, but the next question comes, what is the mechanism by which this diversity actually takes place? And those studies came from another famous biologist, a geneticist, who instead of gazing around the world, looked in his own garden, and that was Mendel. And Mendel showed that there were certain rules of inheritance, and those rules apply both from generation to generation and the way species are formed. But the third component is chemistry. Understanding the structure, the thread of nucleic acid, DNA and RNA, which links all of this life form, and cellular function, means that the metabolism of a cell, the structure of cells actually have common origins, and by looking at unicellular organisms or small insects, worms, and so on and so forth, we can understand about all of life. And that wonder is there in your own garden, in your own backyard, outside in the dirt, in the muck, in the beauty which surrounds us. And taking that and putting it under a microscope is incredibly transformative because it links all life on Earth through a shared chemistry which we know about. That observation with new technologies which allow you to look at the chemistry really can change the way we look at the world and make extraordinary discoveries, but also teach us about the rigors of actually looking at questions driven by our curiosity. That's what Manu has made and his team have made possible. They've taken what was hitherto inaccessible to individuals. Microscopes are typically what you found in a school lab or a college lab, gathering cobwebs, but you didn't look through them to see the spiders. With Manu's microscope, you can look through and look at spiders anywhere. So I won't take more time, we're already running a bit late. So, Manu.
Sounds good. Can everybody hear me? I just have to say thank you so much for hosting me and all the people that made it to Delhi, especially DBT. We're hosting this for a couple of days. And one of the things that I wanted to do was do an informal talk to just get to talk to enough number of people. So before we start, can I just have a show of hands of how many people here were at the workshop today? Okay, so maybe 10% of you actually ended up at the workshop. And then what is the division between arts and sciences here? How many of you are training to be scientists or are enrolled in science courses? Okay. How many arts, literature? There's no artist at all? I'll try to convince you that there is a link between art and science that you should be thinking about. Okay, so Jim, am I connected? Can we dim the lights on the stage for a minute? Can you all see something? Is there a way to dim any lights here? Is this visible to you at the back? Okay, perfect. So, I was thinking about this talk, and I was asking myself a very simple question, which is, if I was in your shoes and somebody was coming by, what would I have wanted to hear? And instead of talking about all kinds of things, I want to frame it in a perspective of a very simple lessons that I care about, that I wish somebody had told me. Some of them will be very obvious to you, and some, it took me almost 20 years of hitting my head on a wall to learn, and I find them very valuable. So we'll essentially start with these lessons and we'll go where we go. Since this is supposed to be informal, if you want to stop and ask me at any point of time, ask questions, it's very valuable. I feel I'm actually making sense if somebody points out and asks questions, so it would be very valuable. If any time you want me to stop, we'll stop. And since they told me I have all the time in the world, I'll talk for a long time, right? No, probably I won't hold you back too long. Okay, so let's start with, these are a few lessons I wish I had learned. And the first one in there is learning how to make observations. So let's switch to the next slide. You know, you all go to the kitchen every day, water spills everywhere. You've all seen this. What happens? Does anybody want to tell me? I take 2 drops of water, I put them on a nice clean surface. I mean, we all have an experience. What happens? Say that again? You can speak loudly. What? But if they're far away, then they can't join together, right? So what would they do? They would evaporate, of course. But what else do they do? Huh? They spread sometimes, and then they stop spreading. Most of the time they don't do anything. Right? That's the observation. That's a very simple observation. You've all made this observation. You put water and it just sits there. Right? In the lab, we made an observation that puzzled us for 4 years. And I want to tell you how fascinating. You can add. I made them yellow. And now, Jim, can you click again? I'm just watching them. Does anybody see something happening? Okay, that makes no sense to me. And it made no sense to me for 4 years. And I'm not kidding. I did this over and over again. Every time, if you go home now, take a clean glass slide. It has to be clean, that's very important. You can clean a glass slide by putting in a little bit of fire, and you put 2 drops of food coloring, and they are talking to each other. Why did that drop on the left know that it should go on the right, and the one on the right knew it has to go on the left? So it took us 4 years of scratching this, and we have partial answers. And I'll share the answer with you, but in the end, that's not why I'm showing this. I'm showing this because observations can come from anywhere. And they are very profound, although you just saw this and you might be thinking, what's the big deal? You should be asking, where is the energy coming from? There's no source of energy that I have put in or showed to you in this system. One clarification I'll make is there's something special. If you do this with pure water, it does not work. Which is what I said the first observation was. It only works if you add food coloring to it. Food coloring primarily has colored dye, but it also has another chemical, propylene glycol. One of the ironic things about this observation is the chemicals don't matter. Water and propylene glycol don't matter. You could take any 2 chemicals as long as they're non-reactive and they don't evaporate very quickly like alcohol. The same observation is universal for binary fluids. We call them binary fluids because they're 2 fluids that are mixed in. So let's go to the next slide. Here is my puzzle. How is it possible that these entities can actually talk to each other at a distance? And to you it might seem like a simple mundane question, you might find an answer, but I'm going to play the next movie and it's going to be happening very fast, so we will play it again, and then I'll come back and tell you a little bit about the explanation. So Jim, can you skip this slide? Let's do math in the end. There will be lots of equations in this talk, so let's just watch this. And hold on a second. Do you think you have audio on this, Jim? Is it possible to connect the audio? There is an audio jack on the left side. There's a magical student of mine who's doing all this magic in the back. So we have to thank him at the end. Okay, so if you have the jack connected, then if you hit play, I'll set up the setting here. You'll be seeing lots of experiments. I mean, it takes 10 minutes to do this experiment. I highly recommend, talking about observation, that you go do this experiment, because it's as trivial as taking some food coloring, you put it with water, you put it on a clean glass slide, and let's watch what happens. Because I want you to jump out of your seat when you see this. Can you? So that's a simple glass slide. You see the length scale. They can climb mountains, so there is a sense of force. They can sense each other if they're exactly the same. They can line up like soldiers. To me, this is the best part. Doing anything. I am just watching the system. If you look inside with a microscope of what's happening, these droplets have a source of energy that we took 4 years to understand, which is an internal flow. So if some of you study chemotaxis in cells, they crawl around, they chase each other, and they have a sense of self. They can detect their own self. But again, it's just a drop of water. So we wrote this paper, and it's very important. I mean, you saw this, but you have no idea how excited we were when we saw this. And to find something so simple, but still remarkably profound, from a perspective of not times on just making novel observations, where we forget how to learn to make novel observations. And this one, literally, I would be lying to you if I said I designed this experiment in my head. Absolutely not. But the only thing that we did at our lab was realize that this is something very special. And the reason it's very special is, let's play the next movie. So if you go to the next slide. How many of you, don't play it yet. How many of you know this movie? How many of you have seen this movie before? One. So to me, in the history of cell biology, this is top 10 movies. You should all have, if you're a scientist, you should have top 10 movies. This is in that top 10. You're watching a neutrophil chase a bacteria. The neutrophil is that funny shape. The little black dumbbell is a bacteria, and the other things are what? The other blobs are red blood cells. Red blood cells don't do anything, but neutrophils inside you keep you alive. Right now, inside you are billions of these little things eating up all the infectious agents that are trying to act on you. So let's play this movie. And it's remarkable for this object to be chasing around a little bacteria. And you know who wins, right? Of course we win. Right there. You know, that's what cells do. But ironically, the process has very fundamental implications to human health, but at that same time, I would be lying to you if I said we actually understand this. We understand the molecules that the bacteria might be generating. We understand that there are molecular motors that are driving motility inside these cells, but we have no idea how billions of molecular motors can coordinate anything, which is a huge problem. If I told you, all of you, with no global signal, that you all talk to each other and figure what we are eating for dinner tonight, you will know what that dinner will look like. It's incredibly hard to build artificially systems that can be synchronous and coherent with no master clock. And on the other hand, you see a system like this doing its thing day in and day out billions of times. And one of the factors that's fun to think about in extremely simple systems of the example that I showed you before. So now we are taking that approach and applying that to understanding cell chemotaxis, where remarkably the molecule that's acting as a signal for those color drop is water itself. Since if I am a drop and I'm evaporating, I am sending out a water vapor from here, which has a radial coordinate system. So the farther you go from me, that water drop signal, the chemistry of cell signaling, you know, it's a very complicated wiring. But you could ask yourself this question: if I start with a simple system that's active to begin with, what we showed in that simple system of water droplets is it's the instabilities that arise in that droplet naturally give rise to cell. And then I could tell you a story to say this is where we want it to end up. This last piece, we are taking this work not just in cell biology, we are taking it in condensed matter physics to understand how magnets work. So one of the things is we're going to abstract what a magnetic domain is and now do the same experiment. So if you click again, Jim, what you're looking at on the top is I now made a grid and I deposited these droplets and I let the system evolve. And you notice a flower pattern emerges on the right. But it's not a perfect pattern. Everybody sees that? It's not perfect. Some flowers are perfect and some are not. That is a telltale sign in physics of what are called frustrated systems. Many magnetic systems are frustrated, and to watch magnets and magnetic domains at a molecular scale requires hundreds of thousands of dollars. And here is an analogy of a system that you could do in a kitchen sink that allows you to explore pattern formation in something as simple as a sketch pen, a food color droplet, and a glass slide. And of course, the evolution of this idea just goes from we want to understand something, but we're also playing. And it might end up in a direction that we are not sure. And you know, this might all be totally fruitless and get forgotten. But it doesn't matter. It matters to me that we made an observation and then we understood something. Because sometimes we're a little too focused on working on something that has an immediate consequences. And every scientist you ask, most of the time, with very impactful work, the first time they were starting, they had no idea whether it will ever be useful for anything. So that's another lesson. That's a free lesson in the middle. Okay, let's switch to lesson number 2. That's just the dynamics. So, okay, value of stupidity. Okay, and now this might sound, can you go back? What do I mean by that? How many of you have read the essay by Michael Schwartz on value of stupidity? So tonight, since I didn't see a raise of hand, I'm the only one. Maybe Jim has read it. I teach it in my class. Tonight, go search Michael Schwartz and value of stupidity in science. And it's a very special type of stupidity. It's the fact that it's okay to make mistakes in science. Most of the time, if you're not making mistakes, you're not pushing yourself to begin with. Sometimes you make mistakes that it hits on you and says, duh, what was I thinking? But on the other hand, if you shun away from your mistakes and you only want a perfect result, most likely you're just repeating somebody else's work. So I'll give you one example personally in the lab, and I didn't want to bring my students' stupidities on the table. I brought mine because I want to tell you I do this on a daily basis. And the stupidity has origin in this word oscillatoria. So if you go to the next slide, here is a remarkable system found in cyanobacteria that my lab is working on, and I've been thinking about for quite a while. What's so remarkable about this system? It's a class of cyanobacteria that have the capacity to nucleate gas bubbles inside a cell. So I'm going to repeat that again. A gas bubble inside a cell. You all know there are organelles, but how can there be air and a little gas bubble that's inside? So what you're looking at up there is a cross-section of a type of a cell, and you see these perfect patterns of hexagons. Those little things are nanometer scale bubbles. And what the organism does, because it lives in the ocean and in the water column, and it depends on light, it synthesizes these bubbles. You know to do what? Can anybody guess? Buoyancy. It goes up and it goes down, and it has the capacity to completely regulate its location, and it's a single cell. So I was fascinated by this system. I've been fascinated by bubbles all my life. I started working on this, and there was a species that I was looking for that does this, but instead of ordering the right species, I ordered something completely wrong. And so if you click the next slide, I ordered a completely wrong culture, and I just said, wow, as a PI, I can't even do this. I can't even get my species right. I felt stupid. I left this bottle. I didn't throw it away. I just forgot about it. It was kept on my bench with sunlight for 6 months. For 6 months, I did not look at this bottle. It was just sitting there. And then I walked back one day, and the bottle had turned into this. Can you go to the next slide? What you're looking at is a surface of the water bottle, and I took photographs of this and I unraveled it. I unwrapped it so that now it looks flat, but it's curled into the surface of the bottle. What you're looking at is another cyanobacteria. It's a filamentous cyanobacteria, and it formed these patterns. It looked like a banyan tree to me at that time. And I was just hooked on to this image. What might possibly be going on? We took these samples and we put it under the microscope. So let's click on the next slide. Hold on 1 second. So now, 2 years later, we actually understand something really remarkable about the system. So I'm going to just play this movie. You should make some observations, and then we will repeat the movie to show you the observations. So let's play this movie. That's what's happening at the microscopic scale. These are individual bacteria that have the capacity to glide. They're interacting with each other, and somehow in those interactions, they start self-organizing. Now you see they've formed a monolayer. And what's really interesting about the system is watch the top left corner. In liquid crystal theory in physics, we call an object like that a defect. It's type of a shape that is preserved, which is a filament that's bent, and another class of filament is trying to cross it but cannot. And strangely, what's so strange about this system is the fact that that defect is present and has memory, it's persistent, although the parts that make that defect don't exist for long periods of time. So I'll repeat that again. Imagine a structure, think of Eiffel Tower, that existed in place and stayed there for a long period of time, but all the little rods and the bolts of that Eiffel Tower were coming away and going in into the system, so the structure is completely emergent. The fact that the structure exists is an emergent property rather than something that was built and frozen in place. There are fascinating number of biological systems that form these structures, but to find something in this system was really valuable for us because we realized we can map this system to a liquid crystal theory. So if you go to the next slide, there are all these patterns that form, and there is a mathematical theory for defining these crystals, and we get to apply hundreds of years of theory of liquid crystals, which is a very well-defined theoretical paradigm, now to a system that forms these emergent patterns and forms structures that are much larger length scales. A fundamental problem in biology is how do you take small parts, say a molecule of tubulin that's only a few hundred nanometers or a few nanometers, to build something like a spindle that is as large as tens or even hundreds of microns. So that length scale gap is very hard to figure out mathematically, and here is an example of a system by applying some very simple rules from liquid crystal theory, we get to explain pattern formation. So this is another example where, of course, it was a failed experiment for what I was trying to do, but at that same time, I got something out of it only because I left that bottle for 6 months. And of course, when I write the paper about it, I'm not going to tell people that's how I got to this question. I'll phrase it in a point that I was always interested in this question, which is always the case. It's perfectly okay when you're writing a story to write the story that you discovered, not the fact that the discovery was made that way. Okay, let's switch to lesson number 3. I want to make sure, how am I doing on time? Because it's very rare that I go slowly. Am I halfway through or even more? Seriously? Awesome. Okay. I can keep going actually. Okay, let's go to lesson number 3. Can you remind me what were the 2 lessons so far? Observations and stupidity, okay? So make sure you remember this because there's a quiz in the end. Something that's been described, there's another essay by Tim Mitchison, what he calls find your own taste in science. Now you might ask, what does that mean, taste in science? Science and taste, what do you mean by that? Your deep interests, because if you're not interested, you'll probably not work as hard as it is needed to in science at a pay that is so low. I'm serious about this. Science, we don't do it for commercial things. You really have to find the class of problems that you will do anything to do that and have that as a career. And in the end, if you're not there for that reason, then maybe you don't belong in science. It's a much of a pursuit that you really have to ask yourself, and only reason to do it if you follow your own taste. I have absolutely no training formally in entomology, but I grew up in India. And every time I would play a soccer game, I remember there would be a cloud of mosquitoes on my head. How many of you have seen that? Okay, all of you. You know, it's a fascinating, not understood phenomena of swarming in insects. First of all, they're detecting you, and the trick I used to play with my friends is I would run close to them, and I would coalesce the swarm, and I would run away, and he has double the mosquitoes I have now. Do this, it works every time. You can apply all kinds of fluid theories to ask, how could these 2 swarms merge together? It's actually a very practical thing to think about because most of the time in swarms are formed by mosquitoes that have mating that happens in flight. So in that swarm are some females and some males, and you can merge them and the ratios will get messed up and they'll divide again. So I can tell you stories about mosquitoes since I was very little because it impacted me, it impacts all of you. They're a nuisance, but they're also very beautiful if you image them. So when I started my lab, I just had this hunch, we've got to do something about this, not knowing what we're going to do. So let's click on the next slide. So I got excited about mosquitoes, and absolutely no training, had never raised a mosquito until I started my lab. And if some of you have raised mosquitoes in the lab, you would realize it's a pain. It's a real pain to maintain cultures, and they're very finicky, they die all the time, although I don't understand why they don't die outside. That has to do with numbers. There are billions and billions of them. So we started raising mosquitoes, and let's go to the next slide. And one question that I was interested in is a technique that was discovered in India by Ronald Ross. How many of you know who Ronald Ross is? So this is the man that connected malaria to mosquitoes. This is the person who fundamentally said a bite of a mosquito is caused by the sporocyte, it's transferred this way. It was done in India. He didn't want to do this work, but in the end he making up a remarkable discovery. He got a Nobel Prize for it. The way he did this is he would sit outside with his microscope, of course, and he would have his assistant sit next to him. The assistant would be bitten by the mosquito. You roll up your sleeves and you sit, and the moment something bites, you catch it. And he puts it under the microscope and he sees something, and one day he saw something swimming in the proboscis of the mosquito. After months and months and months of trying, and this was a technique, it's more than 100 years old, and this is how he mapped where there are mosquitoes that have diseases and where we don't. So now, come back 100 years later, we were in Uganda and starting to think about, could we do something about this problem to know where are the mosquitoes, which species there are. There are more than 3,500 species of mosquitoes, only a few of them carry diseases, but as deforestation happens, completely new viruses that we don't even know exist are crawling out of the forest because these mosquitoes are finding new human hosts. So it's very important to know where the parasites are. It's almost like a drone flying around that could inject you with drugs and all kinds of parasites, but we don't even know what they are. So I went to the field. So if you go to the next slide, and we use a method. So this is just a map that exists. Most of these are computational maps that people have predicted to say where these species are. Many of these methods are mathematical, but the measurements that were made more than 50, 60 years ago because it's very difficult. Imagine if I asked you to map the entire city of Delhi and you went around catching mosquitoes, and ironically, if you switch to the next slide, that's exactly how we do it. So I can pay somebody, and sometimes I sit, I roll up my sleeves. This is how you would do what is called human baiting. For many diseases, this is almost unethical when you think about dengue, because the second type, the second serotype you get, it might be deadly. We don't have any drugs for it. And you catch mosquitoes, and I would start this assay around 6 p.m. in the evening and go on till 10, mosquitoes depending on different time. I would get maybe 20, 30 mosquitoes, I would bring them to the lab, crush them all up, do PCR maybe, and I have data points that are 20 or 30 sample points. And that's not good enough if you're ever going to map an entire continent or the country of India, or even understand how diseases are spreading. So we started thinking a little bit about what could we bring to this problem. As somebody who's never trained in entomologist, we started raising mosquitoes in the lab, and we make a really, I call it a fun discovery that's now panned out to a point where we are starting to apply this tool in the field. So let's go to the next slide. And the discovery was made by a grad student, Haripriya, in the lab. She started culturing mosquitoes, and they started dying. She would grow them again, ramp up, and they would die again. Until one day she said, maybe what I'm feeding them is not good enough, and she went to a kitchen and got Jello. How many of you know what Jello is? It's like a sugar water. It's a very American thing. You eat it with all kinds of things. But it's essentially a cake of sugar with a lot of water content in it. And she started feeding, and the mosquitoes recovered back because they need sugar for nutrition. But she noticed something very interesting. Every single piece of that Jello had a tiny, tiny, tiny drop of saliva in it. Every time the mosquito bites, it bites as a bait, and then I could start doing processing to really get to a point where we could do large-scale mapping. So let's switch to the next slide. And Jim, can you drag this video a little bit? Since I already explained to you the project, maybe I'll switch to the point where it's interesting. So if you play it, this is a doodle sketch that I made. You don't need to see this, but it's a lot of fun. So just drag it a little bit, and I'll tell you when to stop. Oh, no, you have to drag the video. So if you bring your cursor onto the video, yeah, so just start dragging. Keep going, keep going, keep going. Yeah, keep going. Okay, so right here. So now you can just play it. So we started making these chips in the lab. These are microfluidic chips. We do a lot of fabrication in which we embed hydrogel layers and we mimic the tissue structure of what it would look like for mosquitoes to feel. Mosquitoes bite these things, we leave them out in the cage, and in every one of these little places, we are left with a tiny drop of saliva. Now, because it's tiny, it's less than tenths of nanoliters, we get to use very, very small volume reagents. And one of the things we're trying to do, very much like Foldscope, as I'll talk about in a little bit, is to make a tool that you could take a sticker, take it outside in your garden, stick it out there for a week, ship it to a central office in a Ziploc bag, and we would process these on a global scale to try to remap where are diseases coming from. How do these parasites in the vectors actually crawl? And another aspect of this to think about is, I don't know if you've been following some of the work on CRISPR-Cas and genetically modified mosquitoes. On one hand, we have a system that could completely eliminate malaria, and on the other hand, we have a system we have no idea what will happen if we release it out. There's a big ethical debate whether you should release a completely genetically modified mosquito if we don't know, because it's a gene drive, you can't take it back. So should we be making a scale change and completely changing the mosquito population if we don't know how these things evolve? And one of the things we're trying to figure out is can we potentially embed barcodes in these systems to watch how these systems evolve and build measurement and surveillance infrastructure in parallel to the genetic modifications that we would care about. So this goes hand in hand in starting to ask a question that sometimes just purely making measurements in the field is very valuable. Okay, so let's switch to next lesson. One point to give here, talking about frugal science, that's the difference with how long it would take and how much money it would cost for a known number of, say, 50,000 samples versus what you could do in a platform that just shrinks it down. So another value here is to be thinking about how do you really make costs that would be effective on a global scale, where you could say you could take Orissa and the entire state, you could map it completely in potentially real time. Maybe in every month it gets updated, and you ask when a hotspot arises, that's where you do vector control. Because it's a huge challenge. We could do vector control, but should I spray the whole of Delhi, or should I be focused on specific locations? Okay, so let's switch to lesson number 4 or 5. I've probably forgotten by this time. And this is sort of my favorite lesson in the context of frugal science, which is some of the portion of what the lab works on. Let's go to the next slide. And I'm going to try to go a little bit faster because most of the stuff that's published that you've already read is along these lines, so you probably know this, but maybe I'll make this case for transition between information and experience. So information eventually should be free, will be free. You can look anything up. You don't even have to listen to me. You could have just Googled some of these things and you could have looked at them up later. But experience is something, as I said, is very costly. If you don't do it yourself, you will probably not engage. And one of the factors that we've been thinking about is how does information and experience connect? So let's go to the next slide. And this connection between going back and forth, because if you had an experience, if you had looked up what was in your backyard, you could go back and actually dig up the papers from the 1800s to say what was described. And the goal is to make this cycle accessible to everybody around the world. So let's click next. Here is a fascinating example of why I call this frugal science. And now I'm missing my demo. I'm looking for a piece of tape. Anybody who was in the workshop has a piece of tape? It's a really fun demo, so... okay, you will just have to pretend. Anybody has a piece of tape? Okay, I might make do with some tape that I have here. Okay, so I have a piece of Scotch tape right here. You could take any piece of tape. I attach it to any surface, and now I'm going to peel it. And you have to listen to the sound it's going to generate, and it might not make any sound. There was a crackling sound. Let's do it one more time. Did everybody hear that? So, and you know, you've all done this before. You peel tape. right there. The moment I did this, there were X-rays emitted by this piece of Scotch tape. Everybody knows what X-rays are, right? The same X-rays that you use to image this piece of Scotch tape and adhesion is emitting X-rays when you pull it out. Does that make any sense? Right? So that's what I mean by frugal science. Sometimes, and this is not my discovery, this is a discovery by actually a Russian group that had published a paper a long time ago, of course in Russian, and it was dug up, and nobody believed them because they said Scotch tape, X-rays, what are you talking about? And then Putterman, a faculty in UCLA, went out, dug the paper. There's an image. Not only does it emit X-rays, it also emits photons right there at the edge. And literally, they made an X-ray machine and took an image of their own finger using that X-ray, where the power source for the X-ray is a motor attached to the Scotch tape. It completely changes the paradigm of X-rays. I mean, go in any rural area, there are no X-ray machines. I mean, you break a bone and somebody touches and says, okay, this should be okay. Because you require vacuum, you require all kinds of things, high power sources. Here's a purely mechanical way to generate X-rays. And I love this example because I think about frugal science is not just about bringing costs down. It's about thinking about completely different ways of achieving what you would like to achieve. And the physics behind this mechanism is still unknown. I mean, we have some hints on what might be going on, but there is not that much that we know about the mechanism. And now there is a company that spun out of this paper that's trying to provide portable X-ray sources to be used around the world. And so that's one of the contexts of what I think about frugal science, that you really have to be on your feet when you're thinking about what it means to do frugal science. It is no less of a science. Intellectually, it's extremely hard when you add more constraints to yourself and say, I'm still going to try to achieve a certain performance with a cost parameter in mind. And not all of you have to think about it. Sometimes the time it takes to get a result is more important, and then you should just go for buy what you can. But frankly, there's a lot of people in the world which we could use right now to tackle problems around the world. We need their intellect, but we are shoving them to the corner to say, if you don't have all this, you don't have a fancy lab, you can't do science. And I mean, in India, I don't have to repeat this to you. How many of you have a lab at home? Nobody. Tomorrow, you should have a lab at home. At least the people who came with a Foldscope, you're going to take it home, you should set up a little corner. I call them science stations. And start fiddling around, because that's when you will make a real observation. Okay, so let's switch to the next slide. Here, I don't have to repeat this here. There are 2 billion people around the world who live in poverty. 1 billion of that population is actually children. If we're not inspiring these kids to do science, think about the world logically, we're going to be in a situation with a completely divided world where one has extreme technology, they can go to the moon anytime they want, and another who don't even believe in germ theory and will never take a tablet or get their kids vaccinated. And everybody knows the world is a uniform place. It would be a disastrous time. So one of the important aspects to think about this is the factor that we need to do something urgently in the world to make sure science communication is okay. We should all communicate what we do. That's important. I can read in a newspaper, but doing science is even more important. And it's not just for scientists to figure stuff out. You can make observations even if you're not a scientist, professionally, even if you're not being paid for it. You don't get paid that much anyway. Okay, let's switch. This is 2 pictures that remind me of why I do this. Jim took these pictures on our field trip. One of them is the left from Ghana. Does anybody see something wrong in that picture of a school? I mean, there are no kids, that's true, but kids come. But what else is wrong? There's nothing there. There is nothing to be inspired with. It's just benches and tables. how do I think about trying to teach and inspire somebody when this is what majority of the schools look like around the world, and actually, frankly, even in our country? On the right side is a hospital that shows up with a van that shows up once a week, and a doctor is either in the van or maybe not there, sometimes somebody else. And a lot of you work in rural communities. There's a couple of people who came from Orissa, and they know the reality of this, that screening people out in the field is extremely important. So when you do frugal science, the tools that you make might get applications in healthcare, might have applications in education, might be used in places where you would never imagine they would be used. Okay, let's switch. Here's another picture, and again, I don't want to belabor this fact, but the reality of the world is we live in a world where infectious diseases still have a huge toll, even for things that we know how to cure. I took this picture accidentally, and it pains me a lot, and I show it every time. It reminds me of something. This is from Tororo in Uganda, where we were doing some field work. This is the highest probability of you getting infected if you were bitten by a mosquito. It's called the inoculation rate in almost all of Africa. And one of the factors is when you come back, I was playing with these kids, I was walking back, and one of my translators said, do you know what they are sitting on? Those are the graves of their brothers and sisters, and they put them right in the house because they don't forget. And it's very important to think about this, because in the end, it's these communities where you have to ask where frugal science might have the most impact. Because there's absolutely nothing. If you tell them, avoid mosquitoes, you must be joking. So you really have to ask yourself, what types of tools do we ramp up? Much broader than just thinking about where we live. You know, planet is a very broad place, and at that same time, this is the reality of the world where, 600,000 people die every year from a disease like this. So let's switch. And this is another example of a gentleman I met. People tell me sometimes human capital is not there. What is the point of making all these tools? Who's going to use them? Who's going to train them? This is a gentleman that I met that did 4 years of microbiology training. He knows how to grow more number of organisms. He probably knows how to order organisms correctly. And he then is placed out in the field. But look at his lab. I asked him, show me your lab, and the picture at the back is his lab. He said, when I don't have a patient, I use that bed as a lab, and of course, he has nothing. He might as well drive a taxi in Kampala rather than be here with no tools to play with. So, human capital comes from when you train people, but when you give them the tools to do their jobs as well. And this is a common thing that many people talk about that they would pursue. You do not go in many of these areas because you can't do anything, because not enough people are trained, but you have to start somewhere to begin with, and there are plenty of people that are trained. So that's the context of human capital. This is a picture that I like of frozen Jim. Jim is a very animated character. He's sitting up there. On very rare occasions he freezes. And he freezes when he gets really surprised. We were actually taking a malaria training course in Nigeria, and Jim saw something in the microscope and he froze, and I came running and I saw it. So if you click the next slide, he saw that. There were red blood cells, but there was an insect that looked like the same size as a red blood cell with legs, and it was crawling around. And of course, it would be a remarkable discovery if we discovered an arthropod that size of a red blood cell. That was not the case. It's just the instrument had gathered fungus and all kinds of colonies of insects were growing in it. But it was in the field of view. It was inside the instrument. And this is a harsh reality that instruments that you want to make for frugal science not only have to perform, but they have to perform at conditions. There is no air conditioner that you're going to put in. What happened with Ebola crisis, with all these $50,000, $100,000 machines that got shipped in, they all needed air conditioners. And believe me, go back 2 years later and find those machines, how they're doing. So you really have to rethink about places that you would like to work and the tools that you would make. Okay, let's keep going. Here is a span of over the last 4 years. We've been working with many different places. Most of our focus on this space has been healthcare, and then science education came along, and now a big thrust is science education. I want to tell you one more story, so if you click, keep going. There are lots of tools that get used, so we work on many tools, inventing new types of RDTs, new types of microscopes, new ways of sample preparation, and all kinds of molecular ways to diagnose things. But a feedback that I got from somebody in Tororo was this gentleman who'd been training healthcare workers for 30 years. I showed him the microscope, we were doing blood smears, and we were looking at a parasite, and his comment was, you know what, we need to do diagnosis under a tree. And I laughed at it and I said, yeah, we could do that, let's go outside. And then we were standing under the tree and doing the same microscopy. And some of you who came to the workshop, you could do imaging under a tree. But then he showed me these pictures, and these are some of our pictures. But if you click, many screening campaigns are actually run under a tree. So it's not just a joke, you truly have to be out in the field. For a large number of diseases, patients will not come to you, you have to go to them. And that's another context of thinking of both in ecology and biodiversity, where you have to search for samples. So let's keep clicking. So this is the context of microscopy. You all know this. Everybody knows who that is on the left? Do you know what Gandhi is doing in that picture? Anybody knows the history of that picture? It's a fascinating photograph. So it's not clear. There are two stories around it. One is he's self-diagnosing himself of malaria, which I think is pretty cool. And the other could be that he's actually looking at a bacterial sample that somebody showed him. I find another thing that's interesting about that picture is, Gandhi had the classic, he shed all his clothes, nothing foreign, but he knew the value of science, and he is using that microscope. And so there is that contrast that I enjoy in that picture that tells me something about this is a man who understood the value of tools. Okay, so let's keep going. Here's a quick survey of microscopes if you missed the last 400 years of evolution of microscopes in 10 seconds. So just click on that. The first one is a replica of a Leeuwenhoek's microscope. They keep going, and you notice they start becoming objects of art. And my favorite microscope is coming up very soon, right there. And you sort of realize, of course you can add capability and capacity to tools, but you're taking something away as well. You're taking away robustness, you're taking away their portability, and most microscopes look like that. We started asking ourselves that if we're going to make a lot of microscopes, we have to rethink. We cannot just put another object on a table that's built with the same manufacturing technologies. So let's keep going. This is how Foldscope was first built. Now there are many different versions. Since some of you built it already, also most of this is published work. I'm just going to skip and just highlight a few things. So let's keep going. There are many ways to use it. One important philosophical thing that we came up early on, we didn't want to make a tool that could only be used with an instrument like a cell phone. And for disease work, we realized this is actually very valuable, but we wanted to make a tool that you can use anyway, and you can augment it if you use another tool, because not everybody's walking around with this. So let's keep going. Keep going. This is roughly the cost. It cost in different ways and forms. It cost us $1. One fun fact here is if you go down, next slide, Jim and I spent almost 1.5 years just thinking about this slide. And 1.5 years of our life, it's just a single flash of 10 seconds right here. But the fascinating things was the analytical solutions for small-scale optics hadn't been worked out clearly. So we went back to the design board, figured out the analytics, and one of the fun facts there is, of course, the magnification. It's inversely proportional to the types of lenses we use, the radius of curvature, and that's why lenses get smaller and smaller as we make higher magnification lenses. And most of this is published, so anybody who got a Foldscope today, make sure you download that paper, and there's all kinds of information in that paper, how to augment the instrument, how to actually figure out how it works and why it works. Okay, so let's keep going. Let's skip most of this. We tested all kinds of design parameters. Way too much data was collected and was needed. Let's keep going. But this is one context of diseases, and one of the factors, at least most of our big effort in the lab, is focused on making microscopes that are specific to diseases. One of the studies that we just finished is in schistosomiasis. That you can see all the way at the bottom. That's E. coli, bacillus, trypanosomes, giardia, that's Loa Loa. There are a couple pictures of malaria, maybe probably not here. So let's keep going. So one point of that slide is you can do a lot with the type of resolution that's already available at this point. And there's always a trade-off that you could say you can keep improving the tool, or you can start getting it in the hands of communities and get feedback. So if you ever invent a frugal science tool, make sure you don't get it to be perfect. Foldscope is not perfect. We're still working on it. Every couple of weeks we come up with an idea and we say, duh, why didn't we think of this 4 years ago? Just a couple of weeks ago, before arriving here, we included a whole bunch of completely different ways to focus lock, and it's an evolving object, but we took it out to the field and we learned a lot, and that's actually a very good lesson to think about. Okay, so let's keep going. There's a fluorescence. Some people were asking about fluorescence. There is a way you can modify it simply to just do fluorescence. So read the paper, that's the bottom line. Jim just conducted a diagnostic study. I wanted to show you one picture from that, so let's keep going. This is the site in Kenya where he went to a school and we were looking at schistosomiasis. After malaria, it is the most important disease from an economic impact. It's called the trap of poverty. And the reason you get it is you touch water, and most of the water samples are infected by this parasite. The parasite is absolutely freakish. You put your hand in and it can penetrate your skin without a wound. That scares me a little bit, but I got excited about that as well. So in the lab, now we actually study how it penetrates skin and how it moves around and how it finds humans. But Jim was more focused on diagnostics, so he went out to the field. Let's go to the next slide. This is the group that he worked with. These are local people. That was an important aspect, to not just the tool we used in our hand, but we find a team that does this. Let's keep going. And here are just some results. One of the fascinating things is you can measure the effectiveness of a tool from specificity and sensitivity, and we were able to demonstrate very clearly that we can use a tool like this for diagnosis, but also we made fundamental changes that were very specific to schistosomiasis. Because you use wet samples, and most of the time some samples can be infectious, we modified the instrument such that instead of you looking at your eye, it actually projects an image on a surface. But now it reduces eye strain, because then if you're doing microscopy for 8 to 10 hours, you don't have to be sitting looking through that hole. Everybody can do it. And then at that same time, the image is seen by multiple people. So we call this consensus diagnosis, which helps a lot. So anyway, fantastic results, but also reminds you the value of diagnosis, because the school that Jim went to, a significant portion of the kids were found positive, although they had all been treated only 3 or 4 months ago. So the fact that the rate of infection is so high immediately reminds you that you really need to be figuring out much more massive scale for treatment. Okay, so let's keep going. Let's skip this slide. Okay, I'll end with this, which is the community side, which is, I came all this way, not just to talk to you and then I go home and you go home. I came to ask for something. And this is sort of the last lesson. I call it the amoeba in the room. It's a pun on the elephant in the room, except elephants but they're not that abundant actually. And very soon we might not have them, unfortunately. And one of the factors that you have to think about, how does biodiversity play out into this? And how do you engage in something like this? So let's click on the next slide. One of the contexts that Vijay had pointed out is if you look at the eukaryotic family tree, and we talk about metazoans and animals all the time, metazoans occupy a tiny, tiny left corner right there. I have a small letter that says metazoans. That is all the animals that you know. The rest of this biodiversity is really spread in unicellular and multicellular, all kinds of organisms that many times haven't been even mapped out to something like this. So we're in this fundamental shift of, although we have all this biodiversity and we have fascinating tools, we don't have enough people actually exploring some of these worlds. And one of the quotes that I like from E.O. Wilson, how many of you know who E.O. Wilson is? A very famous ant biologist, just an absolutely incredible person. He discovered the most number of species of ants on this planet. Somebody asked him, what would you do if you got another career? He said, I'll take a microscope, and now I'll do the same thing for microbes. So that's sort of an invitation to you. If you find that fascinating, it's a completely open field in terms of really getting to know ecological context of a huge biodiversity of where we come from. And again, you have to reflect back to the fact that I don't know what that knowledge will lead to. Many of these things might actually be parasites. Many of them might give you insights for drugs that you would take and apply them somewhere else. But the fact that we have this, I mean, it's kind of like saying you watch the solar system and your solar system is your focus and the Milky Way is in the background. Think about it. The scale of the Milky Way and the solar system is exactly the scale that we are talking about here, which is the fact of this unexplored biodiversity, and then something that we focus on is stuff that we can see, and also mostly animals. Okay, so let's keep going. One factor for us was, as academics, we write papers. We wrote this paper, Jim and I were scratching our head. We could have written the next paper and then the next paper. We said, for this time, we have to do something different. We need to get this tool to people, and we're working hard to get this tool to people. But if you go to the next slide, we turned the lab into a factory. We built all the manufacturing facilities in the lab. I would buy pizza for anybody who would come and help out in the manufacturing. We built first 50,000 units. And without this paradigm of who knows what people will do, we just started shipping them to anybody who would ask for it and do one single experiment with them. So let's keep going. This is the thing that we had announced. We initially announced 10,000. We ended up scaling that up to 50,000. Let's keep going. And they're around the world. This is one example that I like because it connects science education and global health to me in a very deep way, which is the fact that sometimes you have to ask a question of where is the root of the problem. This is in Tanzania. This is a group of Maasai kids that we work with for a long time, looking at connections between hygiene and science education. And if you click the next picture, this is what you see outside their window. I wish I was in a school like that. They are actually inside Ngorongoro National Park. It is the cradle of life and absolutely incredible biodiversity. If I want to work on lion poop, I will have to jump a lot of hurdles to get lion poop. Here there are kids that are walking around and they're stumbling on it. But at that same time, one of the factors we wanted to ask, if we teach them germ theory, could we change their behavior about sanitation? Very simple idea. If you all taught kids how to wash hands and to wash hands every time they eat, we would cut into half or more of all the death toll that many of these diseases have. So one of the factors that ends up happening is you can keep singing songs, but it doesn't. Observation is the one that makes that connection. So this is an ongoing study, and if anybody is fascinated by social sciences and how would you actually conduct this experiment. I'm not trained as a social scientist. I don't know how to measure behavior. So we're now working with economists and social scientists to try to figure out a metric for behavior measurement to apply that to Foldscope, for example. So let's keep going. I think let's skip this. You all know how fun Foldscope is to use. There was a kid that for the first time discovered that ants sometimes have bubbles inside them. But in the scale-up side, so let's keep going. The instruments are around the world. This is the map of every single place we have shipped these tools. And every dot could be hundreds of microscopes. There are 130 countries. And to me, I'm happy that India is represented okay. But just, today we mapped up 200 more people on that thing. And my hope is that you'll actually use the tool. Because otherwise it's just a waste of effort and it just sits on your shelf. But if you use the tool, you suddenly join this very diverse community, as diverse as the biodiversity that we're excited about exploring. Right there is an island in the middle of nowhere, close to Madagascar. It's not mapped in, but we just shipped 1,000 instruments to Madagascar to a group of kids that are exploring all kinds of parasitic diseases that are found in this very simple primate that people are thinking of making a model system. So I could tell you stories after stories after stories. One of the things we would like to do is to have a community that's connected. So let's keep going, and to connect this community, we made a little website. This is the site that you should register, and if you click on that, actually Jim, maybe you could drag it. It's a movie. If you just make another click, and then just drag it, because probably I want to finish soon. It keeps going. There are thousands of posts, and I could stop anywhere, and then suddenly there'll be a post from El Salvador, and suddenly from US and India, and to me, this is a different way of doing science. So of course I spend time in the lab, but when I come home, I read all these posts. Lax is up there, who's one of the persons I call Foldscopes super users. You become a super user when you get addicted to a Foldscope, when you can't be found without it, where you're posting all the time, where you're sharing. And then I get to have scientific commentary with a broader group of people than I would ever do in my lab. And to me, that is fascinating. And at that same time, I mean, you are all studying science, but there are other people who are not, and you could have a dialogue with them. So it's this very strong plea that I have, that at least the people who participated, make sure you register and actually make a post. This is not Wikipedia, so it's not about being right or wrong, but just making observations. Okay, let's keep going, and I promise you I'm going to finish. Translations. If anybody is in the room who would like to translate Foldscope instruction set into their local language, send me an email. Let's keep going. Since all this is posted on the site, there are all kinds of discoveries that people make. There's a zombie spider that somebody found from Peru, of a fungus that infects the brain of the spider to do whatever the fungus wants to do. There's fascinating source of neuropeptides and things in that fungus itself. This is a map of all kinds of species and the tree of life that has been mapped so far. It's very, very small compared to what we would like to do. Let's keep going. Okay, maybe let's play this video. And of course, you all watched videos like this. This is data. I would have done a live demo if I had a connector here. But this is the type of data that I collect with a Foldscope sitting in a corner of the sea. Does anybody know what that needle is? Anybody has a guess? Watch right there. Of course, they're spear-like objects. What do you think they are? Diatoms. So it turns out, diatoms are not just these pieces of glass. When they're alive, they crawl and walk. And one of the factors is nobody knows how they do that. And so you can just make an observation and you stumble upon a question that nobody knows the answer to. So that's the easy part. But then the hard part is dedicating yourself and taking time to figuring out. And this is the quality type of data. Let's actually slip this movie. Anyway, I'll end here. This is what we would like to do. In some amount of time, try to bring it to a point where every single kid in the world would be walking around with a microscope in their pocket. I don't know what that would do. A lot of people ask me, so what's the point? Maybe I'll do it in the very end. Yeah. Actually, that way I stop talking, because I'm talking in that movie. I hate listening to myself. But this is an important thing. It's also not a trivial challenge. Just to put it in perspective, it's 2 billion kids around the world. We could take many structures to build something in which we go to a toy company and give this patent and have them manufacture, and of course they would go where the market is. But to me, the market is the biodiversity. Biodiversity is spread around. So how do you do it in a way where equity and equality is as important, and how do you scale it such that, what is the point if I sent out tools and they never got used, like maybe 10% of the people in today's workshop who would never touch their Foldscope? So one of the challenges is that I need you guys to ramp up to a point to be the ambassador that you would actually train, and we call this a multiplier effect. And to me that is the only way to scale an idea, because the human capital that's needed to bring science to people is far more costly and far non-trivial than a product or an object. So that's sort of a plea to you that think a little bit about how would you engage the community around you and grow this community. Okay, this is my last slide. So let me just thank some people who worked with me and some of the people who made some of the Foldscope stuff possible. That's Jim Sabalski. He just graduated. You all saw him. This was his PhD thesis, thinking about frugal science. The first time we were posting this and we started asking, many years ago, people were wondering, what are you going to do? And now nobody asks us a question. And many other groups around the university and other places really care. So that was a fantastic transformation. There is Tom, who was visiting, who is a postdoc in the lab. Saad, who couldn't make it because of visa issues. Mary, who's here. George, who is the workforce of the website. Just 2 hours before the workshop was about to go, the website went down. I was calling George. It was middle of the night. He wakes up, fixes up the site, and it's very important that we can make something like that work. Laura, Matthew, Mark, Rachel, and many, many others who've joined the community. But then the biggest thing is the Foldscope community. The two super users, and one of them ended up coming, is Lax. And so after me, he has the most number of posts. Maybe Lax, you beat me now. I should check. But then there are thousands of people that we have to thank. And then finally, DBT to making this possible, having us come down here. If it was that I was just coming and talking, to me, that would have only been a drop in the ocean. But the fact that we were able to work for several hours, I saw data that you guys could collect, and you're going to go home. And how many of you got the micro certificate so far?